Welcome to Spring LIB.
Hi everyone, my name is Eric and let's answer problem 11. Now in this question, we are going to be focusing on eukaryotes and specifically the DNA of eukaryotes. Now there are several factors, including proteins and enzymes, that are involved with the DNA of eukaryotes. Specifically, this can be storage or even DNA replication. In this question, we are going to be going over what the DNA is wrapped on. And let's go through each answer choice to explain what is the right answer. So first off, let's start with A. Now A is histones. Histones are proteins that DNA wrap around to be condensed into chromosomes. So if this is our histone protein, then the DNA will be tightly coiled around the histone, and this will be able to tightly pack the DNA so it can be condensed into a chromosome structure. So A looks to be the right answer, but let's go over the other answers to make sure. Now B is polymerase. Polymerase is the enzyme that adds nucleotides in DNA replication and even in transcription. So what happens is if we have a DNA and we are having nucleotides, then the polymerase will go ahead and add nucleotides to match complementarily, right? So polymerase are the enzymes that can add nucleotides to a DNA sequence. So polymerase is not the correct answer for this question. Now C, we have single-stranded binding proteins. So single-stranded binding proteins. Now these are the proteins that actually will hold together the DNA in certain parts. It will actually keep it straight because DNA will naturally coil, like in this picture here with the histones. So single-stranded binding proteins bind in specific spots, and they hold the DNA in a straight linear fashion. So C is not the correct answer. And finally, D. D is a sliding clamp. Now, a sliding clamp is a protein that actually helps hold the DNA with the polymerase together. Okay, so it makes sure that the polymerase and the DNA actually stay together. And as the name suggests, it will slide along, moving the polymerase down the DNA. So sliding clamp is not the answer. Our correct answer is A, histones. Histones are the proteins that the DNA is going to be wrapped around so it can be condensed into a chromosome. I hope this helps explain, and thanks for watching.